If you're a teacher working with middle school or high school students, it probably won't come as news to you that cognitive faculties are still developing during this period of life. Let's talk a little bit about the changes in brain structure and cognitive function that occur during childhood and adolescence in the normal course of human development. Although we can't directly measure things like synaptic density in adolescent humans, we do have access to structural MRI data, which can quantify the volume of gray and white matter. Postmortem tissue provides snapshots of some of these values as well. We can then combine these measurements with information from the more detailed physiological studies possible in other model organisms, especially non-human primates, over their developmental trajectories. Functional MRI data allows us both to look at activity in different brain areas and to examine correlations in activity between these areas. These correlations are likely to reflect the strength of connectivity between areas. Here's the overall picture which has emerged. First, we should note that humans are born with almost all of the neurons that they'll ever have. There are a few places in the brain where new neurons are created throughout life, most notably in the hippocampus, but in most brain regions, all the neurons that will ever contribute to our behavior are there from day one. Even though the number of neurons is not really changing, the brain does increase in size during development, reaching 95% of adult size around age 11. Since new neurons are not being born, this growth is driven instead by changes in neural connections and synapse number, and by glial cells, especially the myelination of axons. Myelin, remember, is that fatty coating wrapped around an axon, which speeds up signal transmission. Perhaps surprisingly, brain maturation and learning are not characterized by universal continuous increases in volume of each brain area, or in the density of synapses within an area. Instead, early in development, the brain produces about twice as many synapses as will be present in adulthood. Synaptic pruning, in which unwanted synapses are eliminated, seems to be critical for normal development. In prefrontal cortex, synaptic density peaks prior to puberty. Pruning then occurs during adolescence and even continuing throughout our 20s. Likewise, myelination of prefrontal cortex's connections to other brain areas begins in childhood and continues well into our 20s. These changes in brain structure during childhood and adolescence coincide with changes in cognitive abilities and executive function. Inhibitory control, or the ability to resist and suppress impulses, the ability to ignore distractors, the ability to switch between tasks, and the ability to sustain attentional focus over time all improve from childhood up to young adulthood. fMRI imaging also supports the common observation that the ability of our prefrontal cortex to regulate our emotional responses is still developing over these teenage years. So, if you're frustrated with your students, keep in mind that their prefrontal cortex is not functioning at full efficiency yet.